Hello everyone, let me introduce you to our project where we use our novel assay EvIPqPCR for non-invasive detection of the tumorous extracellular vesicles for pancreatic cancer detection. So going to the basic idea why we exactly use extracellular vesicles in our research, and first of all, I would say extracellular vesicles are a great biomarker reservoir for the cancer detection. So basically, it contains all needed material for the research. Current translational research and methods mostly focus on the single marker, so they're definitely complex. Most of the time, it's invasive, it's super expensive, requires relatively large sample volume, and it's time consuming. While our assay requires relatively low sample volume, it's non-invasive, easy to use, less time consuming by avoiding long-step DNA isolation procedure, and most importantly, inexpensive. This brings us to the main purpose of creating a rapid diagnostic method to detect cancer, and more specifically pancreatic cancer, by using tumor-derived extracellular vesicles directly from the patient's serum. Our research mainly focused on the collective attribution rather than single factor by analyzing mitochondrial DNA inside of extracellular vesicles. In order to prove our hypothesis, the tumor cells and EVs are mitochondria rich. As you can see, for our experiment, we choose 3 pancreatic cell lines, which are HPNE, non-cancerous cell line, MIA, cancerous cell line, and PANC, another cancerous cell line with different morphology, and tested them at the cellular level. As you can see, all 3 graphs represent a cycle threshold that has been taken directly from qPCR machine. In this procedure, we use a novel technique called duplexing, where it can quantify 2 probes, both mitochondrial DNA and nucleus DNA, in single reaction. This increases efficiency and accuracy of our assay by reducing the cost of reagents and false results. And as you can see, this result represents a high linearity correlation between cycle threshold and DNA levels that has been used in our case. And it's obvious from the graphs that mitochondrial DNA to nucleus DNA ratio stays almost equal in the HPNE cell line, while in MIA and PANC cell line, mitochondrial DNA was highly elevated compared to the nucleus DNA. The same procedure was later tested on the EV level, and it shows that mitochondrial DNA to nucleus DNA ratio in MIA and PANC was highly significant difference compared to the HPNE, so it proves our hypothesis that tumor cells and EVs are mitochondria rich. So now we successfully proved our hypothesis, and the next question is how to make it useful, easy, and fast in the clinical usage. And this brings us to the idea of creating an important assay that calls EvIPqPCR. So basically a combination of extracellular vesicles, immunoprecipitation, and qPCR. So we already talked about extracellular vesicles and why it is so important in our research, and now we can move to immunoprecipitation procedure. So IP help us to precipitate only tumorous EVs out of whole solution, and by solution I mean a serum sample, by using antibody-antigen binding. So on the surface of the tumorous EVs, there's a special antigen that perfectly fits to the antibody on the surface of our magnetic beads that we use in our IP procedure. So this drugs only tumors EVs down, and this target EVs then tested on the qPCR. And first of all, we conducted in vivo study on mice by dividing into two groups, mice without tumor and mice with tumor. And as you can see, our EvIPqPCR assay showed a significant difference between non-tumorous group to the tumorous group. And next, pilot cohort study was conducted by involving healthy control donors, pancreatitis patients, and pancreatic cancer patients. And as you can see, our assay could perfectly distinguish pancreatic cancer patients from healthy donors, and moreover, it could distinguish pancreatitis patients from healthy donors. And next, correlation study NRC curve was conducted. So correlation study was compared to different stage and grades of the tumor and CA99. So CA99 is the most popular clinical prognostic tumors marker. As you can see, all values showed negative correlation, which indicates that our assay goes more as diagnostic assay rather than prognostic. While our C-curve helps us to determine the diagnostic potential of our assay. So the closer number to 1, the highest diagnostic potential of our assay. As you can see, our assay definitely shows around 0.88, which is definitely close to 1. So as a conclusion, we confidently say that EvIPqPCR goes as a potential diagnostic non-invasive screening for pancreatic cancer detection. Thank you.